We seek to better define the functional map of odor space across glomeruli of the dorsal olfactory bulb by functionally defining glomeruli in terms of the odorants to which they have maximal sensitivity, as well as their relative sensitivities across a carefully selected odorant panel. To this end, we screened around 200 odorants at a range of low concentrations, using a high-throughput olfactometer and an ultrasensitive reporter of neural activity genetically expressed in all olfactory sensory neurons. Our results define threshold concentration ranges and generate consensus high-sensitivity odor maps of OSN inputs to the olfactory bulb. In our experiments, we used a novel olfactometer for high-throughput odor screening, the design of which is illustrated here. Next to it, we show an example of 12 odorants expressed in random order 3 times each to obtain continuous GCAMP6 signal traces from dorsal glomeruli, that is, in OMP iris TTA mice crossed with a TITO GCAMP6 mice. Below, we have an example response matrix of 57 dorsal glomeruli versus 151 odorants. The odorants screened encompassed a wide range of chemical space and included odors from several different chemical categories defined by functional groups. For example, carboxylic acids, aldehydes, esters, and pyrazines, among others. Here we show examples of delta F over F odor maps generated from 4 different experiments. At low odor concentrations, these maps are highly sparse and consistent across animals. Because of this, we are able to identify glomeruli that are the same across experiments, based only on their responsiveness to their best odorant and approximate location. Comparing the response spectra of these identified glomeruli shows consistency across animals. So far, we have been able to do this for over 30 glomeruli. We screened a large set of odors encompassing a wide range of chemical space and included odors from several different chemical categories. By generating delta F over F maps, we can see the specificity of their response. Presenting these odors at nanomolar concentrations reveals very sparse mapping of odor identity. If we zoom in on a few of these maps, we can see some interesting features. For example, we can see that odors that are very similar to each other, such as the ones shown, activate distinct glomeruli, but glomeruli that are very close together. So, activation of these glomeruli by each respective odor is very specific. In this example experiment, we show that the selective activation of 11 domain 1 glomeruli are activated by odors in the picomolar to nanomolar range. Because of this sensitivity, we designed our odor panel to include odors ranging from approximately 0.1 to 1,000 picomolars in dilution. The sparseness of the olfactory bulb response can be illustrated in an example dataset. At low concentrations, the accompanying lifetime sparseness and population sparseness values are very high. Empirical CDF plots of lifetime and population sparseness for 4 such datasets show that most sparseness values range from 0.95 to 1. Here we show CDF plots for experiments in which the odors were presented at concentrations that are 10 times higher. However, we still observe very high sparseness values, ranging from 0.9 to 1. Finally, we created average glomerular tuning by normalizing and ranking glomerular responses, then taking the average of each rank. These show that the response curve of glomeruli on average is very narrow, in both our low concentration odor set and one that is 10 times higher. Overall, this shows that glomeruli and OSNs are highly sensitive and very narrowly tuned to their best odorants, especially in such low concentration regimes. Finally, we created maximum projection maps of glomeruli with maximal sensitivities to chemically similar odorants. We can see that glomeruli with maximal sensitivities to similar odorants are spatially clustered together on the dorsal olfactory bulb surface. However, these appear fragmented and have some overlap. Overall, this supports the idea that there exists a chemotopic map of odorant sensitivity across the olfactory bulb. By screening glomerular responses at near threshold concentrations, we defined consensus functional maps that are consistent across animals. Our results suggest that OSNs are extremely narrowly tuned and sensitive to their most effective odorants. In addition, glomeruli with maximum sensitivities to similar odorants are spatially clustered, supporting that there exists a chemotopic map of odorant sensitivity across the olfactory bulb. As a result, some glomeruli can be identified across animals solely by their most responsive odor and approximate location. Overall, in these concentration regimes, odor representations appear much more sparse, and glomeruli, and therefore olfactory sensory neurons, appear much more narrowly tuned than generally thought. So, there may be less of a role for combinatorial coding strategies in these regimes. Future studies may build upon this work by using it as a platform for designing experiments that probe particular glomeruli and for linking particular glomeruli to odorant receptors.